From the National Institute of Justice's Expert Systems Testbed Project Series, the following presentation is the Technical Capabilities, Concordance Study, and Examples. The NIJ Expert System Testbed Project, commonly referred to as the NEST Project, was launched as a technical assistance project to the forensic DNA community in May of 2005. The goals of the project are to evaluate the commercially available expert systems designed for the technical review of single-source data produced by forensic DNA laboratories, and to report the results to the forensic DNA community. This podcast represents Part 2 of the presentation, Overview of Concordance Study and Visual Introduction to Positive Control and Allelic Ladder. Your presenter is Rhonda Roby, a technical consultant for the NIJ.
In this part of the presentation, we will summarize the evaluation performed with samples for allele concordance on multiple systems. A concordance study was performed that represented more than 500 unique samples for each of the 4 chemistry sets: Amplistar Identifiler PCR Amplification Kit, Amplistar Profiler Plus PCR Amplification Kit, Amplistar Cofiler PCR Amplification Kit, and PowerPlex 16, all separated on the 3100 genetic analyzer. We chose these systems based on a survey we conducted in May 2005 of offender samples that indicated that these are the most commonly used systems for processing offender samples. The check marks represent the different comparisons made for each kit. For example, the results in manual review were compared to the results in GMapper ID, FSSI cubed, and True Allele System 2 for Identifiler. 16 different comparisons were performed with more than 500 samples per comparison. Over 8,800 sample comparisons were performed. The sample sets differ for each chemistry kit used, with a total of over 2,000 samples evaluated. All samples used in the concordance study represented complete profiles, and the data were manually reviewed by qualified forensic scientists. Data was subsequently processed using GMapper ID, FSSI cubed, and True Allele System 2. We evaluated 620 samples in Identifiler, 540 samples in Profiler Plus, 504 samples in Cofiler, and 547 samples in PowerPlex 16, which results in 2,211 different samples that were evaluated, and resulted in over 45,000 alleles that were evaluated in the 3 different expert systems. This table summarizes the results of the 3 expert systems when compared to manual review of the data. An incorrect call is when a peak is assigned the wrong repeat allele number or is not identified. The rule firings are indicative that the locus requires the attention of the analyst, that is, human review. The top number in the table represents the total number of alleles that reported an incorrect allele call and a rule fired. For example, with Identifiler, we looked at over 11,000 alleles in GMapper ID, in FSSI cubed, and in True Allele System 2. In GMapper ID, 49 incorrect allele calls were made, but they fired a rule and brought that particular locus to the attention of the analyst. Whereas with FSSI cubed, 2 incorrect allele calls were made, and in True Allele System 2, 3 incorrect allele calls were made. But I want to make sure it's really clear that these incorrect allele calls are not wrong in the final analysis of the data, because the analyst was then responsible for human review of the data, and they were able to clearly see that there was something going on in that particular locus. The bottom number represents the total number of alleles that are incorrect and no rule fired for that locus. However, a rule did fire in a different locus. So if we look at the data for Profiler Plus, where we evaluated over 9,800 alleles, there was 1 incorrect allele call that did not fire a rule in that locus, but it did fire a rule in an adjoining locus. The same thing with FSSI cubed. There was 1 incorrect allele call that did not fire a rule in that particular locus, but it did fire a rule in an adjoining locus. If we look at the data for Powerplex 16, you can see that we evaluated over 17,000 alleles. In that, in GMAPR-ID, there is a large number of incorrect allele calls that fired a rule in a particular locus. And in comparison, you will see that FSSI cubed only had 24 incorrect allele calls that fired a rule, and in True Allele System 2, we had 11 incorrect allele calls that fired a rule in that particular locus. What's really important to point out here is that the quality of the data affects the way that these particular expert systems can evaluate and manage the data. FSSI cubed and True Allele System 2 were able to compensate for the raised baseline and the spectral pull-up that was observed with this particular data set. In summary, we've evaluated over 45,000 individual alleles using the 4 different chemistry sets. And these 45,000 alleles were evaluated in GMAPR-ID, FSSI cubed, and True Allele System 2. In GMAPR-ID, there were 88 incorrect allele calls that fired a rule, and that is excluding the numbers that you saw and I discussed in Powerplex 16. And there was 1 incorrect allele call that did not fire a rule in that particular locus, but did fire a rule in an adjoining locus. With FSSI cubed, we had 39 incorrect allele calls, but they fired a rule in that locus, and only 1 incorrect allele call that did not fire a rule in that locus, but did fire a rule in the adjoining locus. And in True Allele System 2, we had 28 incorrect allele calls that fired a rule, brought that to the attention of the analyst, and we had 0 incorrect allele calls that did not fire a rule. What all of this data shows us is that these expert systems work. GMAPR-ID, FSSI cubed, and True Allele System 2 all produce accurate allele calling capabilities. When they fire a rule, all that means is it brings that rule to the attention of the analyst, and then it is the analyst's discretion to evaluate each of the rule firings and make the appropriate changes if they are required. What you're not seeing in this table is that there were multiple rule firings that occurred, but not all of those rule firings led to incorrect calls. They just showed the analyst that the data did not meet the laboratory-defined criteria. In the following section, we're going to be looking at different rules that fire for the 3 different expert systems using the same sample through those 3 expert systems. This section will provide examples of rules that fire in the visual display of each expert system. The technical information and rule firings I am about to discuss are only examples of the many features of each system. This presentation is not an exhaustive overview of all the features, limitations, and technical abilities of each system, but is focusing specifically on rule firings. Training from the respective vendors will be necessary to become knowledgeable about all of the features, limitations, file structure, and technical abilities of each software system. Results obtained from each of the expert systems on the same data sets are presented to demonstrate the different rules that fire. Here is an example of what the positive control 9947A looks like with Profiler Plus and GMAPR-ID. In GMAPR-ID, you get a display of the different color separation with each locus identified above that particular allele call for that locus. And you can see that the allele calls are made here. In this display, you're looking at the results of the positive control 9947A with FSSI cubed software. The results for FSSI cubed have a different appearance than what you have seen with the capillary electrophoresis instruments. What occurs in FSSI cubed, it takes the information from a previous software, and the software that we used in this evaluation prior to using FSSI cubed was to export information from GMAPR-ID. We exported information with respect to base pair size, peak height, and peak area. FSSI cubed takes these 3 data points and actually draws spikograms, or stick models of each of the peaks associated with the results from the capillary electrophoresis instrument. I want to bring your attention to the different information that is provided to you in FSSI cubed software. For example, if I blow up this region, you will see that there is a red bar right here. This red bar is indicating this is the locus that I am providing you information on. And so for this locus, we're looking at D3. It is showing us the results of a 14-15 allele for the D3 locus. What you can also see here is that there is a little peak that is prior to the 14 peak. That is a stutter, but it is not being called in this particular display because that stutter has been filtered out appropriately by the software. If I move down and look at another table that is available here in the FSSI Cube display, this area gives me the major designation for the particular locus that is highlighted. In this example, we have the 14-15 allele. If I, as the user, wanted to modify the allele calls that the software had made, I can actually make the allele changes right here under these pull-down menus. I also want to bring to your attention this table. I told you that FSSI Cubed receives data from other software systems. And so it receives the data. All it wants is the base pair size, the peak height, and the peak area. All of the algorithms that are in FSSI Cubed take this information and evaluate the different loci in this particular sample. I mentioned earlier that the 13 peak had been filtered out and that's the stutter, but you can see that there is information for the particular peak that was carried over from the other software system. Another area I want to bring your attention to is this area over here for the rule firings for FSSI Cubed. We will be looking at rule firings for multiple examples following the positive control. If we had a particular rule firing, we would see the name of the rule firing in this area, and we'd see the information with respect to those rule firings here. I also want to show you a couple other features with respect to FSSI Cubed. Up here in the upper right-hand corner is information for mixture interpretation. They have another feature within FSSI Cubed called iStream for mixture interpretation. As you can see, there are no designations made here because we did not evaluate this data under iStream. We're not looking at mixtures, and the data that we're evaluating is only for single source samples. Another area I want to bring your attention to are these arrows that you can see in FSSI Cubed. With these arrows, what you will do in using it as an expert system is that you will click to the next sample that actually had a rule fire. So if all your data, you have good clean data set and you only have 3 rules that fire, let's say, in a batch of 96 samples, it will skip over all the samples that passed your laboratory defined criteria. Here's a display of the positive control 9947A in True Allele System 2. This is just one display of many displays that are available in True Allele System 2. But I'm just showing you here that in fact this is the same locus, the D3 locus, with the same allele calls, and you can see similar information from GMapper ID and also FSSI Cubed in this particular display. So for the D3 locus, you can see that it's calling the 2 major alleles, a 14 allele and a 15 allele, and the stutter peak that we saw in FSSI Cubed is also seen here in True Allele System 2, but it too has been appropriately filtered. Here's a display of the allelic ladder in GMapper ID for PowerPlex 16. It's a pretty busy slide, but you can see that all the loci have all the alleles that are actually called for this sample. Here's the result for an allelic ladder from PowerPlex 16 on FSSI Cubed. FSSI Cubed, as I described earlier, uses base pair size, peak height, and peak area to make the spikograms. In this example of the Penta D locus of PowerPlex 16 in FSSI Cubed software, spikograms are represented for each allele in the allelic ladder. If you can see over here in this region of the software, you can actually see the number of ladders that were run in this batch of samples. There were 3 ladders. And you can also see that it tells you immediately that two ladders in that batch actually passed. And then this particular display of this locus of this allylic ladder, the software also tells you that it passed here. Additional information that you can see in FSSI cubed is information on the allylic ladder. If I blow up the table that is available at the bottom, you will see that you have the base pair size, the peak height, and the peak area associated with each allele of the allylic ladder provided in this table. FSSI cubed takes this information and averages the base pair size and produces a composite allylic ladder for use for the batch of the samples in association with that particular run. This particular sample fell below our defined criteria and our threshold, and therefore a low peak height was defined. Also, you can see that we had an overall genotype quality of a medium value because of the low peak height associated with this particular sample. Again, the expert system brought this sample to our attention. It did make the correct allele call, but it is up to the user to accept or reject this result. Okay, now we're going to be looking at another sample in GMapper ID showing a different rule that has fired. In this particular sample, we have a rule that is fired called low peak height. The low peak height rule has fired because the particular allele for this sample is below the defined threshold for the software. As you can see over here in the left-hand corner, where this peak height is approximately 50 RFU, we have defined it as 100 RFU for this particular allele. In True Allele System 2, for the exact same sample, we can see that the quality value here is 0 again. With this quality value of 0, we want to look at the rules that have fired. And in this particular example, we have two rules that have fired: low homozygote and low signal. As you could see in True Allele System 2, as you also saw in FSSI cubed, it tells you that it's a low homozygote, whereas in GMapper ID, all it told you is that you had a low peak height. So you gain more information in FSSI cubed in True Allele System 2.
This has been an overview of the concordance study, as well as a visual introduction to positive control and allelic ladder. In the next podcast, Rhonda will conclude by presenting visual examples of rule firings in each expert system. She will be showing the identical sample process through each of the systems and discuss the different rule firings. The content of this podcast was funded by the National Institute of Justice, which is part of the Office of Justice programs within the U.S. Department of Justice. The National Institute of Justice, or NIJ, is the research, development, and evaluation arm of the U.S. Department of Justice. NIJ has numerous programs that support state and local crime laboratories across the country.